Hello everybody. Good afternoon one and all. Myself, Dr. Chetna Rajaguru, welcome all of you to the last day, sixth day of our national level lecture series. Dr. Kale sir is giving us his essence of his experiences of life since 5 days. Now, without wasting a moment, I will hand over it to Dr. Kale sir.
Yeah. Thank you very much, Chitra Madam and Valentina, for kind words again. Today we are on the this is our last day in this particular lecture series. This is our last day, and I thought that I thought, is somebody speaking in between? Okay. Today I will begin with 3 episodes. Hello, I can hear some background noise. Please mute your mic. Who is speaking? I don't know who is speaking. Because Valentina, Pankaj and Chetana, all they are got a mute mic, so that there is no problem with them. It must be other participants. So please mute your mic so that I will not get disturbed. I will begin with 3 stories. I know that students like stories. These are real stories, and the first story is of a person called Dr. Alexander Fleming. I'm sure you must have heard his name. And moment I say Alexander Fleming, somebody will say he was responsible for penicillin. But do you know that he was also responsible for finding lysozyme? The two of the most important discoveries which Dr. Alexander Fleming did, they were accidents. In fact, Dr. Alexander Fleming was a very untidy person. That's what he used to claim also. He used to agree with also. There was another gentleman in his lab who was very tidy. His lab was very tidy. But Alexander Fleming's lab was not very tidy. He often used to get contamination. And once he got a contamination, he tried to check it with his that contamination bacteria. One of them, one thing we have to accept. He had very innovative approach of looking at the things. He was never perturbed by the contaminations. In fact, whenever he used to get contamination, he will start thinking that why these bacteria have come there. Um, his famous quote is that one sometimes finds what one is not looking for. One sometimes find one. What one is not looking for. When I woke up just after dawn on September 28, 1928, I certainly did not plan to revolutionize all medicine by discovering the world's first antibiotic or bacterial killer. But I suppose I exactly did that. Now, this was regarding penicillin. But much before the discovery of penicillin, the lysozyme, which is very potent enzyme, which is present in the mucus and in the tears, uh, which actually degrades dissolves the bacterial cell wall and kills the bacterial cell wall. It was also an accidental discovery. So he actually, once he discovered that the tears and mucus has this ability to dissolve the bacteria, he made his colleagues shed tears for next almost 3 weeks, and they used to use onion. Onion cutting was done in the lab so that they will have the uh, tears, and then he used to collect those tears. So sometimes the colleagues got so fed up, they said that अभी हमारे सब आंसू खत्म हो गए हैं, and अभी आपको देने के लिए हमारे पास आंसू नहीं है. Then he began with some lemons. He used to squeeze lemon in the eyes, because if you squeeze lemon in the eyes, that will get the the tears to kind of induce tears. So that induction of the tears he made, and then he found lysozyme. Then in 28 September, he found one contaminated plate. The plate was containing the Staphylococcus aureus, and this Staphylococcus aureus, there was a zone of inhibition. So then he tested various things, and he found one fungus growing there. And this fungus he isolated, and he called it as Penicillium rubrum. And then it was changed to Penicillium chrysogenum. And then again in 2011, it was reclassified as Penicillium rubrum. Of course, Penicillium, many species, now you must be aware, the notatum is there, chrysogenum is there, which actually produce a lot of penicillin. And this was the first incidence of a antibiotic in the history. Probably Dr. Alexander Fleming was not aiming at discovery of penicillin, but he did discover penicillin. And but till you will be surprised, from 1928 to 1944, this discovery was really not noticed by the world. Then once in some other lab, Dr. Flory, he was Dr. Chen was trying to study the structures of various antibiotics, and he found that penicillin has got some wonderful ability of, I mean, being an antibiotic, it can really control many of these gram-positive bacteria. So then he reported this to Dr. Flory, who was the head of the department. Then together they planned some experiment. Dr. Fleming visited this lab. He was very modest. He only suggested that Fleming, the penicillin name should be given to this. So that's how the penicillin came in picture. And in 1945, the what you can call as the medicine in 1945, the Nobel Prize was awarded to these three people, uh, mm, Alexander Fleming and mm, Flory and Chen. Uh, that's how the discoveries happen. In 2005, if you try to go into history, in 2005, uh, there was a Nobel Prize awarded to Dr. Barry Marshall and Dr. Robin Warren. Now, why I'm telling you this story? Because in 1980s, I am sure some of the professors who are of my age, they must have heard the word ulcer, and I'm sure younger generation also must have heard the word ulcer, peptic ulcers. Sometimes there will be holes into your stomach, and I mean, it can go to that extent, and sometimes even peptic cancer can take place. So this peptic ulcer was the one which was investigated by these two gentlemen, Dr. Barry Marshall and Dr. Robin Warren, and they discovered that a particular bacterium, Helicobacter pylori, it was responsible for causing this ulcer. Now, where is this Helicobacter pylori? It is actually a normal flora, and it doesn't grow in other places other than human intestine. So it's a kind of obligate in that sense. It is actually not a parasite, but as far as the environment is concerned, it is obligate. So this discovery that a bacterium causes the peptic ulcer was something novel. This was somewhere in 1951, 52, and then they received the Nobel Prize much later, 2005. I want to tell younger generation that these days you immediately look for some kind of award or something. Usually these Nobel Prizes, youngest person who got the Nobel Prize was probably Dr. Watson in medicine, who got it at the age of 26. But generally the recognition comes much later, because the mm, invention or whatever discovery has been made has to get established, has to get transformed into a product, and then only it can get your appreciation. But these all these Nobel Prize winners, if you look from 1900 to 2020, there are now more than 120 years this Nobel Prize is given. So there must be every year it is given to two or three people in medicine. Very rarely it is given to a single person. But if you can calculate as two, so there are 240 Nobel Prize winners approximately. And I have the actual number from the Nobel side. But why I'm trying to tell you all this is the Nobel Prize was never their aim. Nobody was really aiming for Nobel Prize, but the passion with which they were doing this uh, uh, work and this discovery, it was really something marvelous. And this is what we have to pick up. There is always a question asked why there are not many Nobel Prize winners in India. India has got probably Dr. Subramaniam, who actually migrated. So probably as Indian, Hargobind Singh Khurana was there, who again migrated from here. The C.V. Raman was actually an Indian who remained in India and he got a Nobel Prize in physics. But J.C. Bose came very close to medicine Nobel Prize, but finally he didn't get it. So why there are less scientists who get Nobel Prize in, I mean, who cannot get Nobel Prize in India? That is the question which is to be probably answered. And my answer to that, I mean, sometimes people ask me that हमारे यहाँ क्या कमी है? तो हमारे यहाँ एक कमी है कि पैशन जो रिसर्च में होना चाहिए वो थोड़ा सा कम लग रहा है. ब्रेन आई डोंट थिंक देयर इज एनी प्रॉब्लम, बट पैशन थोड़ी सी कम है. Maybe there is a bias in Nobel committee also. Now people say that some of the Indians came very close to Nobel Prize, but they were not awarded because of the skin color. To some extent it may be true. Maybe some committee members at some stage might have that color feeling in his mind, but generally in science I can tell you that it doesn't happen. When have met so many foreign scientists, I never found that bias in their behavior, in their stayed with some of them for a long period. There was Dr. Fuel in Germany, and the way he treated me for almost 9 months was like his son. There was never a question of any ego or the superiority complex that they are the best and we are not the best. ऐसा कोई मुझे feeling कभी नजर नहीं आई. So I really don't give to that extent to the bias of color. Maybe at some stage some people might have that feeling in the mind, but overall it doesn't affect. The overall what it affects is somewhere the facilities which are there in our, now fortunately UGC, DST, DBT have really put a lot of money in the form of various projects in our laboratories, and they have found that they have made sure that I visited in between a place, I mean university in Ranchi, which is in Jharkhand, and I was astonished to see the type of analytical instruments in that I mean Ranchi University. They had probably the most ultra-modern instruments which were not even in BARC. So I was told that the grants are being given by this UGC, and as I told you yesterday, that now at least you cannot say that there are no facilities in India. You want to get sequencer, the sequencers are available. You want to get the HPLC liquid scintillation counter averaged model, it is available. JCLSC, it is available. GCMS, it is available. LCMS, it is available. So all these very ultra fine or ultra modern equipments and analytical techniques are available in India. So let us hope that we keep a pace of science with that. And that's why my suggestion to all of these college teachers who are listening to me is that the college teachers, they should make sure that one seminar uh, these college teachers should give, and that seminar uh, every year should be somewhere on 5th or 6th October when they declare the Nobel Prize. And this Nobel Prize explanation why the Nobel Prize was given, because these days net information is so fast that the actual prize is being announced there. You can watch it here. So you can certainly learn what for this Nobel Prize is given, and then make a note of this and make a kind of documentary in your own college. And this, I feel that it should be done in every college for every subject. The Nobel Prizes are given for five different disciplines. Now I think six disciplines. Economics is also added. The physics, chemistry, medicine, peace, and literature, and now this economics. So we should make a kind of inspirational program that why this year's Nobel Prize is given, what is the underlying principle behind the experiments which were done by these Nobel Prize winners. When did they do the experiment? One more example I will take this year, 2020. How many of you remember which scientist got the Nobel Prize? If you have got a regular seminar given on that subject, I am sure 5 out of 10 students will definitely remember the Nobel Prize. One out of the four, five, those students which are actually uh, mm, remember this may get inspired, and even if 1 in 1,000 or 1 lakh Indian students get uh, inspired, I think we can achieve a lot by putting this type of inspiration. Also, in food sciences, they don't give Nobel Prize, but in the World Food Organization gives a World Food Prize. This World Food Prize is very important. It is as good as Nobel Prize, and Dr. Swaminathan has got this World Food Prize in the beginning, which was, in fact, it was installed by Dr. Norman Borlaug. So we must have such inspirational people in our country. We must have such inspirational students in our country, and for that, this program is important. The 2020 Nobel Prize was given for hepatitis C virus. If you see that medicine virus, the medicine Nobel Prizes, many of them are for microbiologists. Already I too mentioned to the one for the Helicobacter pylori and the other for the Alexander Fleming's penicillin. It was actually based on microbe only. And this is the today this year's also Nobel Prize was given to Dr. Altar, Dr. Houghton, and Dr. Rice. And these three, they actually solved a puzzle. It's a nice thing to explain the thesis or explain the Nobel Prize in the form of puzzle. What was the puzzle? Puzzle was started in somewhere in 1970s, even in 60s. Dr. Bloomberg, who got a Nobel Prize for hepatitis B in 1976, he was working in 1960s in the National Health Institute for, there were a lot of hepatitis cases. They were not hepatitis A cases. Hepatitis A, you know, the jaundice, which is a very common jaundice where the bile is not, bile accumulates in the body. That's the jaundice, it is caused by hepatitis A, and this was known. But there were a lot of cases which were actually transmitted not through water, and he was working on that. So he found hepatitis B, and then there was an Australian antigen which was found to be similar to this, and then they developed a live vaccine using that Australian antigen, and the hepatitis B was conquered. So he got a Nobel Prize in 1976 for this. But while he was working there, Dr. Altar, who was working in the same lab. He was a young person that time. He noticed that even some of the cases which Dr. Bloomberg was looking as the hepatitis B were not actually hepatitis B. So he started solving this puzzle, and he thought that there must be some other virus which is responsible for that. So then he took up this, and then this was the National Institute of Health in Maryland, that is in USA. So they showed that the blood from these hepatitis patients would actually transmit the disease. They took the blood from the hepatitis C. I mean, this now I know it is C. They took that virus. Now we know that it is not me, all of us, that it is from the hepatitis patients, and he transferred it to chimpanzee. And chimpanzee started showing the similar symptoms. So that infectious agent was shown, and that infectious agent was taken. These various tests were done, and it was shown to be a virus. Now, what to call this virus? So they called it as non-A, non-B virus. And after that, the work was taken up. Now, this puzzle is there. So the puzzle is solved. They say, no, the jigsaw puzzle is there. You have to fix up those blocks at various places, and then at some stage you will solve the puzzle. So this first block of identifying the virus, identifying the agent. Now, once the identification was done, now identification of novel virus was a priority, and this virus eluded these experts for almost a decade. For 10 years, they tried in different means. There was no, they were not able to isolate. That shows the insistence of persistence, high importance of persistence, that we must not leave the problem. We must keep on looking. We know that there is a problem. They knew that there is a hepatitis C patient, and now we know it is hepatitis C, non-A, non-B patient, and they knew that it is a virus. Then why it is eluding? So almost a decade, different doctors tried this, different researcher tried this. Then Dr. Michael Houghton, who was working in a pharmaceutical firm, which is very common, I mean, famous Chiron in Europe, he undertook the arduous work needed to isolate the genetic sequence of the virus, and Houghton and his co-workers created a collection of DNA fragments from nucleic acids found in the blood of the infected chimpanzee. So he went to the infected chimpanzee and tried to isolate those DNA fragments. Of course, majority of the fragments were from chimpanzee, but they predicted that some would be derived from unknown virus. So on the assumption that the antibodies against the virus would be present in the blood taken from the hepatitis patient, investigators, I mean, Houghton and his colleagues, they took the patient's sera to identify the cloned viral DNA fragments from the chimpanzee, and then a comprehensive research was taken, and one positive clone was found. They must have analyzed thousands and thousands of the clones. One clone positive was found, and that further work showed that the clone was derived from a novel RNA virus belonging to the flavivirus family, and it was named as the same family as other hepatitis virus. It was named as hepatitis C. So now the second block of the jigsaw puzzle was put into place, and the first it was identified as virus, the second it was identified as hepatitis C. So what is the problem now? You can declare that it is the hepatitis C virus which is responsible for causing this unknown disease, which was there in Bloomberg's hospital. But no, there is a Robert Koch's postulate that was to be proven, that Dr. Robert Koch had given the postulates to, before declaring any pathogen, you must reinfect that with, I mean, a healthy individual with that pathogen, and if it really creates that disease in the healthy individual, then you can call it as the infective agent. So that was the third block of the And this puzzle was then taken up by Dr. Charles Rice at Washington University at St. Louis, along with the groups working on with RNA viruses. He said they noted a particular characterized region at the end of hepatitis C virus genome, and suspected it could be the important for virus replication. Then he observed genetic variations in the isolated virus samples and hypothesized that some of these might be hinder virus replication through genetic engineering. He generated an RNA variant of hepatitis C virus that included the newly defined region of viral genome, which was devoid of inactivating the genetic variation. From there, he went and showed that yes, this was causing the disease in chimpanzee again, and that is how the puzzle was completely solved. So, genetic engineering, that's what yesterday I promised you to speak something about genetic engineering, something about genetic modifying, modified food, genetically modified plants, genetically modified animals. And this is what I wanted to tell you, that this what is the significance of this Nobel Prize discovery. The Nobel Prize, which 2020 Nobel Prize, that hepatitis C is a landmark achieving in the ongoing battle against viral disease that is there. Because right now we know how we are fighting with coronavirus and what are the difficulties. Now, after listening to this, you may start wondering when actually we'll really identify and prove that corona is responsible. That is not needed in case of corona, because corona is known for these problems in human being, causing different types of flus. The right from simple flu which gets cured within 3 to 4 days. The second is that SARS, MERS, that is found in the Middle East, respiratory syndrome, and then in the SARS. So that is a kind of already some work has been done. We are only looking for the medicine, but it identify. I mean, importance of the work with Dr. Rice, Dr. Altar, and Dr. Houghton is that you should be persistent. You should never leave the problem unless you get the answer to that problem, and that's why the thanks to their discovery that we uh, actually could identify hepatitis C. Now that there is hepatitis D also, hepatitis E also. So the mission now, if you look at the World Health Day this year, the WHO had declared it as the hepatitis free next decade. We should make this world free of hepatitis virus completely. A, B, C, D, E, F, whatever they are there, they should be eliminated. So this also was cured, and this is another significance. Hepatitis C was cured by raising hopes of eradicating this hepatitis C from the world population, like what smallpox was done. Edward Jenner developed that vaccine, and smallpox is eradicated. When I was young, there used to be slogans that to find out a hepatitis, a smallpox patient, and you will get a reward of 1,000 rupees. At time we were young people, we were not understanding why this was such kind of why somebody should pay for finding a deceased person. So once our teacher had explained the meaning of this, that there is no more smallpox cases. So if you find a smallpox case, that will be giving a trigger to the medical machinery of the country that in your country this disease is not eradicated, so you must take care. And that's how the this mission was done. In this competition was taken at the insistence of WHO in all the countries in the world, and that's how the smallpox was totally eradicated. So the antiviral drugs or the antiviral vaccines, although they are known from smallpox and polio time and rabies time, the hepatitis belongs to a very different kind of group like coronavirus, which are RNA viruses, and getting a vaccine for RNA virus is rather difficult. So we are looking at various controversial news coming from Oxford vaccine, Russian vaccine, and we are really quite confused that what are these? Sometimes they say now we have solved the problem, we are vaccine at doorstep, and tomorrow we will get vaccinated. The next day news comes, sorry, that vaccine is no more effective. So these are the problems involved in the RNA virus vaccines, that the instability is very, I mean, glaring, and because of that instability, getting a stable vaccine is going to be a tough, I wouldn't say it is impossible for a human being with so much of advancement in science. Probably you will be able to find out something for that, but I'm sure that as of today, it's going to be a very, very difficult situation as far to find out a vaccine. So you have to learn from this, and this is what my another homework to all the, especially the staff member, is and this staff members not only for biotechnology or microbiology, but for all life sciences, for physics, chemistry, that please call that assembly on, say, something like 10th October, when all the Nobel Prizes are declared for that year, and let the eminent people in your locality talk about these Nobel Prizes, explain to people. One thing I can tell you, that in Germany, when I was staying, I learned a lot. Sometimes I jokingly say that I learned probably the entire science in my life from my mother and from those 10 months in Germany, because the everything what my mother did and everything what the German people did was science. They were able to explain each and every step to me, like what my mother used to do. So the in the Germany, I found that every weekend there will be a kind of meeting of the citizens with the various scientists in that city, because Germany is one of the best examples of the decentralization. If you look at the population of the German cities, it has not increased or decreased over a period of time. The growth rate is 0%, and this same growth rate is there in villages. And why that is happened? Why the dream of Mahatma Gandhi, that we can uh, a decentralized structure of a country, is much better if you have the small hamlets which are self-sufficient in their all the activities. Then you need not crowd in the cities. And you have seen there were so many predictions for Mumbai, for Delhi, for Calcutta. Fortunately, maybe because we are untidy people like Alexander Fleming, and because he was untidy, he could get some of those very important researches made through the accidents. I also feel that sometimes, because we are so untidy in our habits, we are so unhygienic in habits, probably our result immunity power is much better. Even sometimes I jokingly say in some of my lectures that if we drink pure water every day, we may suffer because we are used to drink contaminated water. But I mean, keep apart from the joke, the point is that it's God's gracing that the corona has not really broken up in that fashion. But that doesn't mean that we should take things lightly. So these are the lessons which we get from the Nobel Prizes. Some of these Nobel Prizes, look at those winners like Max Planck and the Chadwick. Many of these they have come up from a very difficult situation. But every time, each Nobel Prize winner's character, of course, there will be many others because everybody doesn't get Nobel Prize. But I am sure there is a good cross section of scientists in this world, and that's why learning science history should be made as compulsory, and that should be taught in a manner in which it should be inspiring. It should not happen that you are forcing a child, or I mean, now these students will be of something like 20, 21 years of old. It should not look as a boring thing for them, but take up some important aspects of and give of those characters, of those biographies. And these biographies are plenty available on your net. Maybe some teacher can take up it to translate into the local languages. Somebody can translate in Gujarati, somebody can translate it in Malayali. I am writing, so many of my colleagues have written on Marathi. I am writing one book on the inspiring characters in science. Around 50 I intend to write. But these make them available as a kind of rapid reader. I remember in our school we had that in syllabus there used to be a rapid reader. What was a rapid reader? Rapid reader is something which will not be told in depth, which will not be taught in the school, but you are supposed to read it, and there will be one small paper on that. Maybe you can add it as a credit course without actually marks, but that should be compulsory. And if we can make them read, I am sure because maybe I'm wrong, but wherever I have gone to the colleges when I inquire, there is not such course taught to them about the history of the sciences. History of sciences is really wonderful. You know, one of these scientists, his name was, you must have read his book, Leonardo da Vinci. He used to write from the right to left, and he was writing in opposite manner. You know, he suppose the word is bold, he will say DLOB. So you have to read it really, and he wrote something like 4,500 pages like this. All his work, which was related to science, he was a scientist. The way he worked in science and the way he wrote his communications, while communicating to Royal Society, he used to, of course, write in common language because then nobody will understand what he was writing if he was. But his records he was putting in the form of that 4,500 pages record is still there in somewhere in Italy in museum that Somerset Maugham has done this type of research. And these Royal Society letters, I think not Alexander, this father of microbiology, Leeuwenhoek, he must have written something like 400 letters to, he will take this scraping of the tooth, observe it, then write it what he has observed. He was not a good in art, so he had kept one person to draw the things he used to explain, and then the person used to draw. So, like that, 400 letters he has sent various things. Then there was a dispute about what he was describing. So, Royal Society had put a kind of Robert Hooke was there in the secretary's post in Royal Society. So, and he was very fond of Leeuwenhoek because his letters he found very innovative, and there was a lot of information coming from Leeuwenhoek. So, when Royal Society asked him to look into the matter, he sent one committee to Leeuwenhoek, and then that committee went and saw all the literature, all the data of Leeuwenhoek, and then they certified that what Leeuwenhoek is telling is right, and that's how these letters were then they found a place in the Royal Society journals, proceeding the Royal Society. I will. My sincere request to all of you is while publishing these days, there are a lot of open access journals. There is a paid, like paid news are there. There are paid papers. Please don't go for that. Paid papers are not going to give you any satisfaction because then you yourself are guilty and you know that you are got the paper. Unknowingly, I got trapped into one such incident, and I was really sad about it. That why I should publish a paper because at the end only they told me that you have to pay this much of money. One paper I withdrew anyway, but one had gone to so far extent that then there would have been a legal dispute between two countries. So then I paid some dollars, but I don't include that paper in my publication list. So this is what is finally important, that your paper should be peer reviewed. If it is not, and that's why your scientific writing should be very good. Many times our papers are coming back saying that poor language. There will be good people in English, and that's where the interdepartmental collaboration is very important. That writing papers, you should never feel below dignity to get your paper evaluated from your colleagues. In fact, if you are fighting with some colleague, the best way is to give that colleague, अरे ये मेरा पेपर जरा evaluate करके दो, दोनों के relation भी ठीक हो जाएंगे, and then he will be try to be as critical, and you will try to solve the problem. So try to get it evaporate. I think Valentina is calling. What is the problem? Yes, yes, or it is going on. Yeah, yeah. I don't find any problem. ठीक है ना? Okay. Sorry, this was a disturbance from the, not disturbance, this was a phone call from the organizers because they had lost connectivity. But I thought, I think that all the students, all the students are connected well, and we can continue. I'll just check it because in one of the student is sitting in next to my
Yes, sir, we can
resident.
listen to you.
I will just confirm and
We can
it
listen
is
to you, sir.
okay, okay, fine, fine. So, because I wanted some kind of feedback. Okay. Yeah. So coming back to what I was talking is that the quality of the publications which is coming from our country, and it is where probably one has to be including all the administrators, principals, vice chancellors, and the people at helm of the affairs. They should take a decision that what kind of peer reviewing system should be generated within the departments. And I am of the opinion that unless you get it weighted, get it self-evaluated. Self-evaluation means not my paper should be done by me, but my colleagues. And that type of evaluation, if it is done, there is a discussion, healthy discussion. And that is another thing which I learned in Germany. They had a weekly meeting. I don't know how many of research organizations or the colleges have got a weekly meeting to evaluate the research which is happening in that college. They had a every week meeting. One person will present, and every 6 months, I remember Dr. Fuel taking me to some interiors of the country. There they will enjoy for 2 days, and those 2 days, the enjoyment will not be there during daytime. The from morning 8 o'clock to evening 8 o'clock, evening 8 o'clock, 12 hours, there will be a complete review of the work. So that 10 minutes presentation is not there. Every person has to present for about an hour, and discussion can go up to 2 or 3 hours. So if there are 12 or 14 colleagues in that department, the process will go on for 2 days for 12 hours. That kind of rigorous evaluation was there, the good laboratory practices were there, and that is how the I'm trying to answer you the question why we are not having the good Nobel Prize winners in our country is because we will have to do this self retrospection, that are we really at par. Maybe some of you will be saying that अरे हम तो बहुत ऊपर पहुंचे हुए हैं, ठीक है आप ऊपर पहुंचे हुए हैं, ये हमारे जो systems है ये system में आप ऊपर पहुंच भी गए होंगे, but is it really standing firm on the foundation, and is that foundation very strong? And for that we must have that self analysis which is going on. Yesterday I told you that we should really look into these various concepts of organic farming, various concepts of genetically modified food, whether they should be. There is a debate going on about the genetically modified brinjals, the whether we should eat those genetically modified brinjals, whether we should have the Monsanto cotton, which is that BT cotton, they call it. The Bacillus thuringiensis gene is incorporated into cotton so that it is insect resistant, but many people say that it is also breaking. Let me tell you that in nature, if you try to do something, there will be a, Newton had given one beautiful law that for every reaction there is a equal but opposite reaction. And nature is not exception to that. So if you want to conquer the nature, nature will again have hit back and try to do the corrections, because for nature you are not a favorite species. For nature, you are one of the species. And if one of the species is getting dominant and is trying to make sure that the other enemies of you are eliminated, nature will try to protect them. So this fight is going to continue. And this is where I always say that while using the pesticides, while using the force against some people, you should never be having that killer instinct. What is your, you should say, like our Santa Ramdas Swami used to say that when you are constructing a new house, the house belongs to you. But at the same time, there are so many small animals which are living in your house. That house belongs to them also. There will be ants, there will be cockroaches, there will be flies, there will be lizards, and they also will say that this is my house because it belongs to nature. You may have purchased a particular plot of land, but finally that plot of land belongs to the Mother Earth, and we are nobody to purchase it. It is just a kind of equivalence which we have created because I have money and I can have this place isolated for me. So we call it as house. But is it really your house? No, it's not your house. After say when you die, that house the grass will grow and in the cows will graze. Nothing will remain of that house. You have seen how the things are transformed over a period of time. There were palaces in Jaipur, there were palaces all over the country because our India was a the several kings were there. At every 10 kilometers, there will be a different king, and they will have their kingdoms. So all that kingdoms are lost now. There are no, even our Mayur Simhasan, which we are very fond of, is no more with us. So what probably the Mughal Badshahs, what Shivaji Maharaj did on Raigad, now there are only some remaining. So this is time. Time always washes things down. What we have to learn from this history is that how they, within his time, how Shivaji Maharaj was great, or how this Queen of Jhansi was, she fought for freedom. There were several of such people who had fought for freedom, how they did it in their time. And nothing is long lasting, but the work which you do is long lasting. That's why you are in field of science, and that science should be long lasting. And while you are living, while you are breathing, what is the difference between you and a common person who has not really studied science? According to me, every person is a scientist because he takes oxygen in his breath, he drinks water which is H2O, and he eats food which is of chemicals, organic chemicals maybe, but they are chemicals. So there is nothing different from a scientist. If I am drinking water and you are drinking water, if my blood group is AB and some of you may be having A or B, what is the difference between two of us? It's only the question of thinking. And why that difference of thinking is there? Because of the culture, because of the education, because of the surroundings, because of the sanskars which have happened over a period of time on various people, from various people, like from your parents, from your teachers, from your school, from the society who are around you. There is a story of two parrots. One parrot, when there are twin parrots, and one parrot goes to the some sadhus or rishis, and the other parrot is taken by some thieves. So these thieves parrot starts talking, he starts abusing whenever somebody comes. Get out from here! Why you have come? I will kill you! Give me your money! That type of thing that parrot talks. The other fellow, the parrot who is with his some good sage people, saintly people, he whenever somebody comes, he will welcome. Swagat hai aapka, aap आइए बैठिए, आपको पानी चाहिए क्या? Now these are twin parrots. The same thing, the mentality you see in human beings also. So your job as a student, your job as a teacher, you are learned people. You have learned a subject, microbiology. You have learned a subject, biotechnology, and it is your job to imbibe that culture which you have studied in your school and your college, and now you are implementing in your research. That should spread. That and for that you should be confident enough. Once I had the discussion that organic farming is good and India is using too much of urea. India is using too much of inorganic fertilizer. So the topsoil layer of our country is in danger. Well, all the argument is right. But at the same time, another argument is if you don't use them, what will happen? Do you have enough of organic manure in your country to replace urea? Do you have means of doing that? Look at the some of the negative aspects of organic manure. I was surprised to see a paper in Science, which was published somewhere in 2000, that the myths of organic farming. You can even look at that for the paper somewhere around 2000 or maybe 1999. In Science, it is published, and it's called Myths of Organic Farming. There, the researcher, and Science is a very prestigious journal, one of the prestigious journals. So, in Science paper, it is written that why I am calling organic farming as myth, because when organic farming was not there, only organic farming was not there. Sorry, when organic farming was intervened. Earlier, it was only organic farming. There were no pesticides, no fertilizers. So, time immemorial, what our ancestors are doing is organic farming. But later on, as the 19th century, the DDT was discovered, and then there were too many insects, especially in tropical countries because of the weather. These pesticides came into picture, DDT came, organochlorine, other things came, HCH or the benzene hexachloride, it came, then there was endosulfan, then there was this like eldrine, aldrine, quite toxic chemicals came into environment. Then there was a novel written by Raquel Carson, Silent Valley, which really opened up the eyes of people. Then they started opposing the pesticide. The green people went to an extent that it should be completely banned. Then DDT was withdrawn from the circulation in first in Europe, then in America, and then in India. In India, tropical country, 45-degree temperature is reached at many places in Rajasthan, in Madhya Pradesh. Actually, the DDT also degrades quite fast there. But once the decision is taken, fatwa आया, सबने follow कर दिया that we want to withdraw that DDT, fine. But these organochlorine, then there will be organophosphorus pesticides came into picture, carbamates came into picture, like your chlorpyrifos, then the monoprotophos, they came into picture, the Savin or the carbaryl. Then that Bhopal incident happened, and then carbaryl was because one of the ingredients which spread in the environment from the union carbide was methyl isocyanate, which is the starting material for carbaryl. So that also, all that happened, and the pesticide changed from one generation to the other, to the third generation. Now there is more emphasis on neem-based pesticides because they want to be organic. So all that happened, and urea was introduced. Then they said that urea will get wipe away the topsoil layer because urea will be instantly taken up by the plant, and then plant will not have any rhizosphere effect and bacteria will die. All kinds of explanations were given. There were practical experiences, whether I don't know whether that was the reason, but practical experiences that urea had really stripped off all the, helped all the salts to deposit into the soils, and the lands became unfertile. Well, partly it may be true also, and partly it is true also. But what is the alternative? What is the solution for that? And then there was one aspect brought in that Science paper, that when these pesticides were there in somewhere in 60s and 70s, the colic cancer or the anal cancer, the peptic cancer, the incidences had reduced drastically because on these peptic cancers or the colic cancer, main culprit was the mycotoxins. And once the this myco means fungi, the fungal toxins, and once this fungus was completely killed because of the various pesticides, the stored grains will not have any of those fungi. So there were no mycotoxins present in the stored grains like wheat and jowar. And because of that, and rice, and because of that, the peptic cancer had drastically reduced. So it was a boon for stopping the peptic cancer. The inorganic pesticides actually helped in reducing. So there will be always a kind of what I am trying to tell you is a plus or minus, and conscious, something you have to be very conscious, and that consciousness is what you are learning. Right now, my dear friends, you are not learning microbiology, you are not learning biotechnology. As I told you again and again, you are learning consciousness. Then to learn this consciousness, your means is microbiology or your means is biotechnology. So what is your role, that through the principles of microbiology and through the principles of biotechnology, you should understand the life values. And these life values will help you in your healthy and happy life. Uh, I may sound to you philosophical. Some younger generation people may say that, सर ऐसा क्यों बोल रहे हैं, ये तो लाइफ है, ये माइक्रोबायोलॉजी है, इसका हमारे जीवन में क्या रिलेशन है? बहुत बहुत गहरा रिलेशन है इसके साथ। जब आपकी आंखें खुली रहती है, जैसे अलेक्जेंडर फ्लेमिंग की आंखें अगर खुली नहीं रहती थी, he would not have seen that contamination or discarded that contamination as in when our labs we are doing some pure culture technique, and if a plate gets contaminated, what is our reaction? अरे इसको sterilize करके फेंक दो, इसका agar melt कर दो, उसको फेंक दो. Now, what is this? This is something. I am not saying that you should look at every contaminated plate, but you should know what this contamination is being caused. So mostly bacillus is the culprit in our tropical area. The contamination will come because of the bacillus. Have you ever checked the efficiency of your laminar flow? The manometer which is there on laminar flow will actually show the pressure which is coming from that HEPA filter, the positive pressure. But manometer has only kind of showpiece in many of the laboratories. They don't know even for what that manometer is kept there. And the red liquid which is present in the manometer, it is going up or down. Nobody really knows the importance of that. If it crosses a particular mark, and that is the Lakshman Rekha. If that Lakshman Rekha is there, it reaches. That means the pressure is now no more coming. The pressurized air is not coming from the HEPA filter. So what it indicates now, it is time to clean the HEPA filters. The HEPA filters are blocked. It is not generating that. What is the meaning of our laminar flow or clean air? It is the positive pressure. The filtered air is coming so that the outside air will not get into the chamber, and your chamber will not get contaminated. That is the principle of laminar flow. So that is laminar flow. So the flow is in one direction. And if your pressure is not there in HEPA filter, then if the pressurized air is not coming, then your room air will come into the chamber. It will get contaminated, and that's why you will get often contamination. So this is what is again the values of science education, again in the form of the good laboratory practices or quality control. You can give n number of names to that. You can teach in n number of forms. These days they call it for PhD. There is I forgot the name. You people have that one paper that for good laboratory practices equivalent, that they should know the research methodologies. They call it as research methodologies. Well, subject is very good. The reason why it is introduced also is very good. How many of you really know research methodologies? And again, I'm not going to take examination of you. I'm not here for evaluation. I'm not here for assessment. I'm here only to tell you that, well, these are important things. And because all these Nobel Prize winners have followed these things more or less to the finest of the letters in that SOPs, they could come to this level. Okay, so organic farming, coming back to the subject, that many times the question is asked where this organic manure will come for such a great country. More than 141 million hectares of land is under cultivation. So 100 million hectares land, if 1 ton of organic manure is also to be provided, 141 million tons of organic farm manure which will have to be given, because a minimum quantity of organic manure to be added in the soil has to be 1 to 2 tons per acre, and we have 141 million hectares. So into 2.5, that much of tonnage of organic manure should be available. Well, organic manure is there plenty. Unfortunately, you don't realize. Me, once I was with parliamentary committee, we had visited Punjab, and we were going into that place called Ludhiana, and there is a Buddha Nala in Ludhiana, and the Buddha Nala is full of only gobar. You know, Ludhiana is a place which is very good for. Punjab has done milk revolution just like Gujarat has done, and many farmers have come together, and those big cattle sheds are there. But unfortunately, the gobar is flowing into the Buddha Nala, and it actually goes in Satluj later. So huge amount of gobar is going. It is getting degraded into the finally nature will take care of it. Nature will put it for recycling if you can't put it. So nature will do it, but then the reduction of methane which is happening, that from gobar, the production of methane, reduction of that gobar into methane, which is happening, and this methane is going into the environment, which is responsible for global warming. So we are losing at many counts. We are losing that good manure in the form of gobar, and we are adding to the global warming because of so much of methane going into this. Simply, you have to put up that entire river, which is Vudanala, and connect it to a big biogas plant and convert it. Many times people ask us in my field that what is the return from this? How much of gas will come? Is my plant going to be self-sustainable? Is my there will be enough funds generated out of this? My dear friends, it is not always money, and sometimes you have to see the recycling. Somehow you have to see the continuation of life cycle. And in our government department, when you go to as a government rule, you buy something, you have to pay GST on that. The tax earlier you were paying excise, you were paying extra, you were paying different types of taxes. Now there is a different taxation structure, and you have to pay GST. Do you question really that why you have to pay GST? Maybe you are getting fed up with that percentage, and so you may be angry that you have to pay this much of GST. But finally, you have to pay GST. These are the taxes, and why the GST is to be paid? This GST is the income for the government, and the income tax is the income for the government, which will take that money and use it, respend it. So it's a recycling which is happening of the funding. So you are taking part of your earnings and paying to government, so that you are requesting government that okay, you maintain the infrastructure, you maintain the roads, you maintain the clean air, you maintain the clean water supply to the city. So all these infrastructure which is required for a healthy life is being maintained by the government. So the government will have some expenditure, and where this expenditure will come from, it will come from your GST. Now just extend that system to the earth, the mother earth, and all this mother earth is supplying is whatever the existing natural sources to you. Where is going to earth is going to get carbon dioxide? Where is earth going to get water? It is from the existing sources also, and these existing sources are to be properly planned. They should be utilized. There will be waste generation. That waste has to be treated. So all that will need some kind of expenditure. Now many times I hear a comparison that in Europe they do everything. Their vehicles run on biogas, and why can't we do it? Well, we can do it. But you know, the biogas plant there costs almost 3 times or 4 times more than the biogas plants in India, because in India at every level you start asking. I'm not talking corruption, that is anyway a different matter, but I'm talking of the actual expenditure. And this expenditure is the moment you say to tell municipality this plant will need this much of maintenance, either they close the plant or they back out, and they say that no, 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 we are not going to use this. We will throw it on dumping yard. So these are the type of mentalities we have to fight. Sometimes when I come back from Germany or from USA, I find that we are 30 years or 40 years behind them in this thinking. Maybe we are using all the medical, scientific developments, but in our thinking we are behind. We cannot think that spend, we should spend money for the wastewater. If we don't spend money to treat the wastewater, tomorrow we are not going to get good water. So some kind of expenditure, that is the GST, is needed, and that's why you should not think in terms of money, but you should think in terms of the producing as much of manure, and every individual can do it. That's what I am trying to tell you. If you look at the dumping yard in India, you will find that 30 to 40% of the waste, or maybe up to 50% at many places, is only the kitchen waste. It can be the cooked food, as it happens in many marriage ceremonies, in seminars. I'm sure you are scientific community, and that seminars, how much food you waste yourself. I'm not going to ask you embarrassing questions, but I have seen this in happening in seminars. So much of food is wasted. We take too much, although there is a buffet system. We take too much of food and then throw it because we are not really concerned about it. So 40 to 50% of our waste is kitchen waste. Out of that, 20% is can be stopped, or 30% can be stopped completely if you take a oath that I will not throw a single gram. And a single person is Aham Brahmasmi, as I told you, because if you are the world, then you should do it. And if you are doing it, the other person will say Aham Brahmasmi, third will say Aham Brahmasmi, then everybody will do it. So you can, all of you together can reduce this 20 to 30% of the food waste, and then remaining 20% is the actual kitchen waste, which you cannot digest. So you cannot eat, like banana skin you cannot eat, the cucumber skin you cannot eat, बैंगन का जो पीछे वाला हिस्सा होता है आप खा नहीं सकते, आप काट के उसको फेंक देते हैं. सो दैट, और कोरिएंडर का जो बैक का हिस्सा होता है वो आप काट के फेंक देते हो. All that can be collected and converted in manure. My personal experience, I tell you, for last several years, even one house with 2 or 3 people can generate 5 kilograms of extremely good manure in your house every month. And think of the country, think of the population of the country. If there are 30 crore houses, 30 crore into 5, that is 150 crore kilograms of very good quality organic manure can be generated in your house, and it is multiplied by 12, that is 1800 crore kilograms of very good organic manure supply is coming from individual residents. Then there will be plenty of organic manure generated in the field of these rice straw, of the wheat straw, of the sugarcane bagasse, and so many things are there which are not actually utilized. But if we can develop means of, and that's where microbiology is, that's what biotechnology is, that try to get it degraded within a period of 1 year, so that that's the, in my kitchen, in whatever waste we are generating, we are converting it into manure in 48 hours, and it's no fairy tale, or it's not a something an exaggerated information I'm giving you. I'm giving you the practical information which do it regularly, and simply lactic culture from your buttermilk, and some good management practices like cutting the waste into extremely small pieces with a lot of passion, and then mixing it while adding, and then not putting anything on the top soil layer. These are simple SOPs for this generation of organic manure. If you are interested, can send an email to me. I will send a detailed procedure, and you can establish this in your house. Then use the same manure to grow some hydroponics or some pot cultures. Grow your own chili, your own coriander, and it's an excellent exercise for you and for people who are in your house. they also get interested once they see that, okay, this is giving us the good pesticide free pesticide, I mean, crops like simple crops like palak, the spinach or methi or those type of vegetable. You can have some runners like your cucurbit family. It can be cucumber, it can be these pumpkins. You can grow them in your balcony if there is a sunlight there. And if sunlight is not there also, you can make some LED light arrangement and do it. So it's too many, I mean, it's a question of as I say always, sky is the limit. There is plenty of things available around us. Only thing, we should be innovative enough to think about it. And by doing that, if 30 crore people are doing this, the dumping yard can be stopped in one day, and India can become extremely clean. So I will like to ask you in the form of a poem that I want to ask you a question. क्या बदलाव आएगा? My name of this poem is क्या बदलाव आएगा? चांद पे तो पहुंच गए थे कई साल पहले, अब अंतरिक्ष तो नया नहीं है, पहुंचेंगे हम मंगल ग्रह पर, शायद उसके आगे भी, आएगी कल बुलेट ट्रेन हमारे देश में, होगी कई सारी स्मार्ट नगरी हमारे देश में. I have seen Rajkot in the name of smart nagari. I want to ask question to Valentina and others from Rajkot. Is your Rajkot really smart? I find Pune in smart city list. I will not vouch that Pune is a smart city. I can see that waste is right now lying in front of me in that building. So how can I say it is a smart city? So my poem goes further. क्या होगी कई सारी स्मार्ट नगरी हमारे देश में, लेकिन क्या बदल सकेंगे हम खुद को? क्या करेंगे कूड़ा मुक्त अपने घर को? डंपिंग यार्ड बढ़ाते जा रहे हैं, कीटाणुओं को बढ़ावा दे रहे हैं, घर का कचरा पड़ोस में फेंक के घर तो स्वच्छ होगा मेरे मित्र, लेकिन क्या सोचा है अभी भारत स्वच्छ होगा? उसी कूड़े से आएगी ना कल के अनाज की बोरी, यही बतला रही ना पदार्थ के अविनाशित्व की कहानी. कूड़ा बाहर फेंकने से जहां दिन की शुरुआत होती है, कूड़ा बाहर फेंकने से जहां दिन की शुरुआत होती है, वहां भारत बदलने की बात हो रही है, वहां भारत बदलने की बात हो रही है, मेरा भारत महान. Are you really willing to change? Are you willing to change from inside? Yes, I will make zero waste, and I told you my concept of zero waste. By giving my waste to the sweeper, that is not zero waste. That is plus some half a kilo or plus 2 kilos of waste every day in your house. It is not zero waste. To make your house zero waste, you should give nothing to that person who is collecting. Instead, you should give him some resource which he can sell. And that is what I am expecting, a good segregation. Isn't it a job of a biotechnologist and microbiologist to understand? Isn't it a job of microbiologist and biotechnologist to take it forward? Isn't it a job of a biotechnologist and microbiologist to develop a good culture and give it to the society? So that's what I am expecting. I expect that you people should take up these matters. There will be plenty of other things in life, but this is your mandate. This mandate is given to you by God because of your subject is so nice. You take the real active culture. Lactic culture is one of the most wonderful culture in the world. It converts things in such a beautiful form. Good idli, good dosa, good curd, good cheese. All this is given to you by microbes. The same microbes are there who can willingly take up your uh, wasted resource from your kitchen and convert it to an excellent manure and provide it to the Mother Earth. You cannot say that I don't have time to cut it into small pieces. You have time for everything. You have time for eating, you have time for sleeping, you have time for enjoying, you have time for watching movies, you have time for watching IPLs. Well, I have no quarrel with that. But isn't it your responsibility also to take up 10 minutes and help your mother or your wife or who is cooking in the house that to connect that waste and not to give it outside, but convert it to a manure? If you can't do it at your home, you can do it at community level. So many of the senior citizens will be supported by you, by your activity, they will come forward and help you also. So this has to come from internal. And all my lecture series is to convert it internally, convert you internally into a good human being. Yeah, I don't want to make you tell you something exciting that when I say inspiring, it is not exciting that every day I can give an idea. अरे आज तुम ये करो, आज एसी का पानी कलेक्ट करो, आज वहाँ घर का ऑर्गेनिक मैन्योर कलेक्ट करो, and then become rich. I'm not here to tell you how to make rich, how to become rich. You may be better people to teach me, because a person like me, I even don't charge for anything. So you may say, सर आप ये क्या कर रहे हैं, आपके लेक्चर के आप 5,000 रुपए ले सकते थे, रजिस्ट्रेशन ले सकते थे. नहीं करना है मुझे वो. I never had that intention in my mind. The question is, if I am born in this world, I owe this world something. If I am using the resources of this world, you know how much of oxygen you use every day? You breathe in 21,000 times, you take 3 kg oxygen, and today corona time, you know what is the value of that oxygen? It is 33,000 rupees for 3 kilos. So every day you are using 33,000 rupees kilo oxygen without paying bill. If like electricity, God says that your electricity, your oxygen supply will be cut if you don't pay 33,000 rupees, you will be taken off from this world immediately, because I don't think you can pay 33,000 rupees per day. None of you can pay. And this is only one element I'm talking. There are 23 elements in your body. If I give you that bill, this bill will be of 5 lakh rupees. Can you pay this bill for 23? Your lunch and dinner may cost 200 rupees or 300 rupees in a restaurant. But that is only the service which he is charging. He is not charging for real element. The real element you have to pay. This is the GST you have to pay. And this is what, as a microbiologist, as a biotechnologist, you must understand your duties, my dear friends, my dear students, and my dear teachers. So this is all exercises for this. So make sure that you are a good human being. You are a good microbiologist. So like our Harivansh Rai Bachchan says, the सब जगह अंधेरा तो फैलता है, लेकिन अंधेरी रात में हाथ में दीपक लेने को किसने मना किया है क्या? नहीं किया है. विज्ञान का दीपक आपके हाथ में है. I'll say last poem of mine and then I take leave from all of you. अब मैं नहीं घबराता. सोच के पहले मैं घबराता था, कहीं मुझमें का इंसान खो तो नहीं रहा है. पहले सोच के मैं घबराता था मित्रों, कहीं मुझमें का इंसान खो तो नहीं रहा है. जब मैं बाजार में जाता था हाथ में थैली लिए बिना, तो मेरे प्रेस्टीज को कोई धक्का नहीं लगता था. जब मैं हाथ में जाता हूँ थैली लिए बिना, और जब भर के प्लास्टिक की थैली या सब्जी लाता हूँ घर में, तो मुझे कोई शर्म नहीं आती है. क्या सिर्फ मेरी प्लास्टिक की थैलियां ही प्रदूषण फैला रही है? सबके थैलियां प्रदूषण नहीं फैला रही है. जब मैं बाहर जाता हूँ आलीशान गाड़ी में, आजकल लोकल गाड़ी में जाना मुश्किल हो गया है, तो मेरे मन में सवाल नहीं आता कि रास्ते के मुसाफिर कैसे चल रहे हैं. ना मुझे लगता है कि मेरी कार प्रदूषण फैला रही है. क्या सिर्फ मेरी ही कार प्रदूषण फैला रही है? रास्ते की सारी कारें प्रदूषण नहीं फैला रही है? जब मैं वातानुकूल कमरे में अपने सोता हूँ कंबल पहन के, तो मेरे मन में ख्याल नहीं आता देश की आधी आबादी कंबल खरीद भी नहीं सकती. कहीं मुझमें का इंसान खो तो नहीं रहा है. जब मैं वीडियो लेने जाता हूँ कोई एक्सीडेंट का या किसी के मौत का, तो मेरे पास आगे जाके मदद करने के लिए समय नहीं है, क्योंकि मुझे वो वीडियो लेके व्हाट्सएप पे या फेसबुक पे डाल के लाइक्स कमाने हैं. मैं सोच के घबराता था पहले कि मुझमें का इंसान खो तो नहीं रहा है. लेकिन आजकल मैं बिल्कुल नहीं घबराता दोस्तों. आजकल मैं बिल्कुल नहीं घबराता दोस्तों, क्योंकि मेरे अंदर का इंसान मर चुका है. मेरे अंदर का इंसान मर चुका है. मैंने अब सोचना ही बंद कर दिया है. नमस्ते, धन्यवाद.
सर आपका ये पोएम है ना वो भेज दीजिएगा कभी भी हम लोगों को प्लीज.
क्या करूं? क्या करूं?
आपका पोएम है ना, आपका पोएम है ना वो वेलेंटिना मैडम को भेज देना, उनके पास से ले लेंगे.
I have actually put there is a book. I mean, I really don't intend to make money, but I have published it on Amazon. I can send you the link.
Okay, sure, sir.
I have got about 80 poems of science.
And sir, if it is possible, आप यो सेम है ना पठन कर दीजिएगा आपकी आवाज में, वैसा चाहिए.
जरूर.
ओके सर, थैंक यू. हैव अ नाइस टाइम.
थैंक यू. There is one more small thing which I wanted to show you. This is the organic manure which I generate from my own house every day. You can see it is completely free of insects. There is a bucket, and then this is a plant. This is methi which I have grown, and this is sufficient for 3 of us. Right now, one of my daughter, niece is there, and my wife. So this is sufficient for us. So this I do it every day. I will make sure that it is done every day. So you can take over, Chetna madam or Valentina.
Thank you very much, sir, for your valuable and memorable lectures, which will surely going to compel us to retrospect. Now, I will request our head and associate professor, Dr. Valentina Umbrania, to reflect upon this national level lecture series. Please, madam.
Thank you, Chetna madam. Thank you, sir. Actually, there are no words to give any overview or summarizing this thing, because sir has highly inspirational talk has been given to us by every day, day to day, and it was so much effective for the motivation actually to rise our consciousness and to perform our mindful efforts to live science, to drink science, to survive with the science, and how to serve our nation and Mother Earth and all over living and non-living on this planet Earth. As it is very much nicely said that Vidya is something which cannot be stolen. We have lots of beautiful words in our Sanskrit. I'm not very much learned in Sanskrit, but few shlok which I like so much. One of them is Nachoraharyam, Nacharajaharyam, Na Bhatrubhajyam, Nachabharakari. Vyaye krute vardhata eva nityam. Every day it is increasing when this vidya धनम सर्व धनम प्रधानम व्यय कृत्य जितना उसको यूज करते हैं इतना हमारी विद्या बढ़ती है. तो सर ने भी हमको इतना सारा विद्या ज्ञान दिया कि हमको पता चला कि हाउ व्हाट इज द पर्पस ऑफ दिस लाइफ एंड हाउ science is inculcated in this thing. As again I remember Sajjanan Lakshanani in our Sanskrit Subhashita, all the deed are done for the paropakara. So sir has done really great paropakara to enlighten the lives of so many students and staff and all interested people who are listening in this channel. Sir has started and gave us from the word that I don't know. When we say that we don't know, then only we are ready to learn something. When our cup is empty, then only we can fill it with another concepts of wisdom and knowledge. So we started with I don't know concept, Kayamat se Kayamat tak, the poem, and then generation of the patriotism, faithfulness, and wish to serve our country. Janani Janmabhumischa Svargadapi Garyasi. Sir has told us that Panch Mahabhut se ye body bana hai, to we have to take care of our air, our water, our soil, everything which is around us. So all environmental factors, how they can be taken care by ourselves, and how we can return to our Mother Earth, so that coming generations can live their life in a healthy manner. Sir has touched actually all the basic science as well as so many life science concepts, like starting from nanotechnology in the difference between laddu and halwa. Sir has taken us to the physics, nanotechnology, instrumentation, microscopy, advancement with the RT-PCR techniques, how autoclave is working, how we are dealing with pressure cooker. This cooker syndrome, sir earlier has also told to us that this cooker syndrome is responsible to melt the glaciers and to give global warming. And then he enlightened us with small is large. He gave us the pathway how to study, how to make focus, how to read between the lines, between the words, between the letters. ओनली शब्दार्थ को नहीं समझना है, लेकिन गूढ़ अर्थ को समझ के उसको जीवन में कैसे उतार सके हम, ताकि वी डू समथिंग गुड फॉर द सोसाइटी एंड फॉर आवरसेल्व्स. Sir has also touched medical microbiology, history of microbiology, so much fascinating facts from the life of the scientist. In this pandemic corona, how viruses are connected to our life. With the chemistry, sir has explained us that being water is an universal solvent. All the body reactions are possible with this universal solvent, and that is the necessity of the water. And sustainability of the environment was how nicely explained to us, showing the model where the ecosystem cycles are running in the automatic manner, where utpatti, vriddhi or lay are in the cyclic manner, in a balanced manner. It is going on in the nature. So many things are told by sir, that kachra kachra nahi hai, it is our resources, how to use our resources and how to make them more fruitful for the not only humankind but also living and non-living on this planet Earth. I highly show my gratitude. I express deep sense of thanks to sir for sparing time for 6 days to be with us for 1.5 hours every day, and to make us clear in our vision for the purpose of this life and for the purpose of getting studied, not only to get degree, but to increase our knowledge, wisdom, and to make it spread among the society and to implement it in our day-to-day life. Thank you so much, sir. Thank you very much. Namaste.
Thank you, Valentina madam, for your kind words. Now, I would request our young and energetic principal, Dr. K.J. Ganatra sir, to express his impressions for the event. Please, sir.
Sir, please unmute your mic. Doctor, your mic is muted.
Oh,
Yes.
yeah. Thank you. Respected Dr. Kale, sir, my colleagues from the microbiology department, Valentina Ben, Jetta Ben Juhi, my other staff members, Jatin Bhai, Nitin Bhai, Pankaj Bechovatia, and other faculties, and all the my dear students and participants who have been logged in uh, to this national level lecture series. It has been 6 long days that I would literally say Dr. Kale has kept us on hook uh, to this subject. Uh, what is very inspiring for all of us, I think, is firstly that, sir, even without a PowerPoint presentation, you we were kept hooked to your talk, sir, throughout all these 6 days. I think it is never heard that one can keep and one can have engaged the participant merely by uh, talking to the audience, sir. Sir, thank you very much. It was really uh, very touching, like and inspirational. Talk. Sir, you covered from the beginning. You covered all the aspects of course, the historical aspects of microbiology, touching the subjects of corona and water conservation and pesticides and organic farming. So I think at the end, uh, for the students, for the teachers, and also for the researchers, I think what sir has given us about the concept, I think this is what is inspiring. That the thoughts put forward, the historical aspects of the subject. I think this is what would inspire us to think differently and to move ahead in our respective subjects. And the most touching aspect was that it is very rare that even I have seen such a person like who is talking through the poems, the very basic facts. So that is very commendable, sir. I've never listened to anyone to make this subject so interesting, sir. Like talking through your poem. And the most inspiring thing is what I think sir has taught us to all of us is that what we have been given, we should give back to Mother Earth, and to be a good human being, to utilize this subject to give to the society, to do something for the betterment of society. I remember on the very, I think, first or second day, sir gave us a very innovative idea of how collecting the wastewater from the like AC generation plants, etc. I think we could never have given a thought to that, sir. It is all because of you that now we could probably move in some different direction with this thought. And this is what this talk, this whole series was all about, sir. So thank you very much, sir. Thank you all of you. All of you, stay safe and happy Diwali, sir. Thank you.
Thank you very much, Dr. Ganatra. Thank you very much.
I look forward to meet you, sir, in Rajkot.
Sure.
After everything is over, sir. We look forward.
I will come. I will come.
Yes, sir.
Thank you very much, Ganatra sir. Now, I would invite our little teacher, Juhi Sheth, for vote of thanks.
Thank you so much, madam. Good evening to all present over here and all the dignitaries present. Namaste and Jai Jalaram from Juhi Sheth, from Jalaram Uch Kelavani Mandal, MBM Science and Home Science College, Rajkot, Microbiology Department. I would like to thank Dr. Kale sir for enlightening us for 6 consecutive days. He has enlightened us in so many ways that we could not even give a thought or haven't we imagined. Thank you, sir, for such an inspiring lectures. I thank the trustees of our college, Principal Dr. Ganatra sir, for his kind support and guidance for carrying out this lecture series. I thank all the staff members of Microbiology Department, Dr. Valentina Umbrania and Dr. Chetna Rajaguru for carrying out this session and giving constant inspiration to do more work. I thank all the staff members of our college, chemistry department and all other departments of science and home science. I will also acknowledge the presence of Pankaj Chovatia sir and Nitin Giri Gosai sir for their technical support and help. Without them, this session would not be possible. Very big thank you to both of them, sir. I again thank all the teaching and non-teaching staff of MVM Science and Home Science College, management of the college, and special again thanks to Principal Sir for providing us the infrastructure. I thank all the participants for joining the session and making it a grand success. I wish that at the end of the series, or at the end of the session, this is not the end, this is the beginning to give a new thought, a new beginning for doing something fruitful towards our Mother Earth, Mother Nature, and our country. As Walt Disney said, the best way to get started is to quit talking and begin doing. So let us all begin doing something good, something useful. Like we should not waste food, we should not waste water, and we should not pollute the air. At least these three basic things, if we start implementing this Diwali, this new year, we can surely give a new start and new beginning for making Earth a cleaner and greener place to live in. Again, I thank all those who are behind the curtains for making this event successful. Thank you so much, everybody. Thank you, sir. Namaste.
Yes.
Thank you.
Thank you, sir. Namaste.
Thank you, sir. Thank you very much. Thank you, all of you.